check you on different so different type of data means the H&E means the nucleus should be stained with purple and cell with pink, and as a PAS means the nucleus should be in blue and sugar should be in magenta, and immunochemistry data should be nucleus should be blue and protein should be brown, and trichrome should be nucleus should be black and muscle should be red and collagen should be blue. So each one should indicate different things like H&E will check you is sick, whether the biopsy is a sick one, or the PAS will go for the where is the sick part, and ISC will go for how sick, and trichrome will go for what sick. So each indication we needed. Commonly used models sometimes go for H&E or PAS only, but a model should learn something means it should see all type of data. That should be the rule, the thing. And I already mentioned that first we get the biopsy, we just cut into thin slices, then we stain, then we want the scanning. So different scanners are available and different formats are available. So like NDPI, SPS, Apperio scanner, Himanshu scanner, lots of scanners are available. So you can see from the slide, there are different views for a same image, like from a top view, you can see everything. And if you can go further, you will get more details. So the scanning is an important artifact of the data. And this is the whole slide image which we are going to give for our model. As we are taking any data set for medical image processing, this is the way in which you are getting the histopathology data through this process. But as a computer engineer, we should be focused on this part only. But the previous part is very important as it reveals the real fact about the data. So the WSI image should be a massive size of a gigapixel image, and it has of a tiling of EC processing and a pyramidal structure. As you can see, the side perspective, like a tissue overview, you can see at a fold level, and a 5x zoom, you can see regions, and 10x, you can see the architecture, 20x cellular, and 40x is subcellular. And if you are going for the scale problem, suppose a single WSI image at a 40x magnification, the resolution should be of 100K to 800K. Then for the file size of 1 to 4 GB. And if you are cutting into patches, like we don't have to feed everything into the model, because as you know, the model is using some CUDA, and the CUDA should have a RAM capability, and the capability should be overflowed by this big resolution image. So we have to cut that into the small patches, and each of size 255 or 256 or 224, like that. And it should have a, from one single image, you will get 150K something number of patches. It should be depends. But this is the way in which your data's nature in a histopathology image. As we knew that a tissue should have a physical space and a pixel space, because when you scan an image, you should convert that into pixel space. So suppose you have a slide image, it's of 40x scan, and your pixel size is sometimes 0.25 micron per pixel. So you will get only the physical size of 56 into 56 microns only. Why it is significant? Because if you are taking a kidney image, it has several tissue parts like Lupo-Henle should be there, glomeruli should be there. So each should have a physical micron space. So we have to understand that in order to detect that, right? So if you are taking a 20x scan, you will get more number of contacts, like 120, 112, 112 micron you will get. So suppose you are scanning at 10x, you will get 224 into 224 microns. So this is the way in which we have to see when you are patching or when you are taking a data, you should see what type of structure you need. Because for a kidney biopsy, you should look for this. For a liver biopsy, some other things. So every biopsy should focus on the inherent biological structure. While analyzing the data, we should keep in mind that what is a pixel space and what is the physical tissue space. Then only we can model the thing. And as we knew to do some pre-processing. As every model, every data, we should need some pre-processing. As considering of different staining techniques already I have mentioned, we have to do a stain normalization. There may be fixed algorithms like Mekengo, Reinhardt algorithms are available. Depends on the data property, we have to identify which type of normalization we have to apply. And there is one more concept like some new algorithms which are telling like you don't have to do pre-process. We will take the noise and everything into the model and we can learn. That is the best thing. But even though now also we do this stain normalization because then only we will get the output which we desired. So that is a kind of tech thing we can experiment on. If people working on PhDs or something, they can go for the normalization techniques like new normalization algorithms they can apply. And the tissue segmentation is an Ustu threshold or other methods like a simple threshold we can apply and we can find out the mask from the image. What is the need? Because if you are taking a WSI image, it should have a lot of white space because the tissue part should be very low and the big outer part should be there. So we have to find out the mask in which we have to find the tissue parts relevant. Otherwise, you are wasting your computation time for processing the background. So that's more important. And that's called tissue segmentation. And there may be lot of algorithms like HistoQC, GrandQC, and HistoSweep. So many algorithms are available. And let more things to come. Because the HistoSweep is the art kind of algorithm because it's developed by a pathologist. They analyze different types of tissues and they find out a filters like the ordinary image processing filters. They find out and they do that for. But the people who are working better in this area is Harvard Medical Science, and they find out a new like grand QC kind of algorithm where they feed the image. The QC should automatically detect the artifacts or the pen markings, etc., from the thing, and they should remove that. So for our pipeline, what should be the need that we have to understand? We have to apply the segmentation accordingly. So in case of a people who are working biomedical, they can go for an ablation like the different types of segmentation, different types of normalization. They can apply and they can find out the result should be different. And another part is like patch extraction or the exclusive filtering, because we knew that once we got a mask, we cut into patches, and the patches may be relevant. Sometimes some patches may not be relevant because they should have some inbuilt tissue space, etc. The space should be sometimes we have to keep and sometimes we have to remove. So that should be the need of the QC filtering inside the patch extraction. That is, the tissue should be greater than 50%. We will keep that, otherwise we will skip that. So this is about the pre-processing pipeline. Then as we go as a computer people, we should more focus on the model development. As you know already, everything like a neural network, it should learn from the weight and the bias. And from there, we are getting the y is equal to sigmoid of wxi plus wxi plus b. And all things learned because of this only theorem that called universal approximation, that it can learn any shape. So this is the basic fundamental, even though if you are going for a big neural network or a small neural network, because of this approximation theorem only, the network should learn the thing. And a few family of CNNs evolved at a time, as these saw from 2012 to 2020, that is AlexNet. It came with a Hindens paper. This is a basic foundational model paper in 2012. What is the innovation is that the Kuskowski actually he learned with GPUs how to do the deep net architectures. In 2014, another team came up with a VGG16 visual geometry group. They have come with a depth and simplicity should be the pattern of developing the CNNs, even though some people are now also using VGGs for feature extraction. And then Microsoft came with a ResNet. This is the hard backbone for all networks now. Even though the transformers, if you give an image, sometimes you should pass to a ResNet to get a good feature. What is their contribution is a skip connection. Why these design choices are important? Because whenever you knew that any neural network should learn through backpropagation. So while you are backpropagating, you are propagating the gradients backwards, right? So the gradients should not lose at any point. So whenever you are training a neural network, you should focus on how much gradients is flowing and how where you are losing the gradient. Because if the layer is too much deeper, when it come back, you will lose the gradients, of course. So this network comes with an idea of this how to the gradient, how should we keep up to the base layer. So that is what they came up with a skip connection. Then DenseNet came with a huge connectivity should increase the retention power, and they came up with dense connection. And then come with the efficient net, they come with a compound scaling, that like some other parameters also used for the scaling thing. And this is what the evolution of family of CNNs came, and they become the backbone of any of networks. Then the real come, that is on 2017 was one et paper, that is the attention all you need. This paper is the most foundational one. After that, 2020 onwards, people will focus only on this kind of uh, models. What is the idea is like already you have understood that transformers, the core architecture. Why these transformers are that much important? Nothing more than only a neural network only, but still how they process the thing, that should more important. Like they have an attention mechanism. What is this attention mechanism? Because we knew that the features are in a feature space and they are called vectors, and how much they are similar. So previously, what people are doing is that they're finding out the similarity, but can we learn the similarity? That asks the question of attention, and that's why the query key value pairs came into play, and they make the cosine similarity and they multiply with the value, and this is what the intuition behind the attention mechanism. So even though if you take any model, even it's a multi-model, uh, image or video or like text or anything, what we have to do is that we have to convert that into vectors, and we have to find the similarity. And for that, we will apply the attention. This learning will make the network to focus on more reliable things. So, applying transformers in 2026 is a cliche thing. Why we have to focus? Because there are different training strategies we have to learn in order to train a network. Because even though if your data is abundant and if you have a network which is of huge size, people because focused on 50 billion parameters, 100 billion, like the exploring more number of parameters. So train the model without losing gradient, without losing the without understanding what learning rate, how can I start? Because that was Karpati told like 3 years to -4, we can start with any network. But we have to find out a learning rate which should make it good. So there are papers like came in ICLR like what type of hyperparameters I can use for training a network. That should be more important for a real world problem. And then 2023 onwards, the people are talking about foundational models. Why foundational models? Because you have different data sources and you are finding about an embedding, and that embedding should be an output from a network. That's called foundational model, and it can be used. This embedding can be used for any type of purpose like segmentation, classification, prediction, and scoring. So these are the four outcomes of any network. Even though if you start from 2009, if you end at 2027 or 2026, also your this downstream task should be something to this one only. Even though if you medical image or if you are taking some other domain also, these are the core problems. And once you make the model, every time you are again training and retraining, that should be a waste of computation. So can we fine tune and reuse? That should be the choice of foundational model. Because once you develop the model, people can use your embeddings and they can train their own model for their own purpose. That is why the idea of foundational model came. Now I'm talking not about 2023. Now reasoning models and agentic AI came, but foundational models are to be the turn representations of the data. Because if I train a huge model for different types of images, and if I get an embedding from that, that can be utilized for the further purpose. That is why actually Gemini and Gamma, a lot of models came into different place, and we can use that model and we can use for our purpose also. That is a clever tricks we have to apply to get the inference from the model. And these are the some of the foundational models in biology, because actually I'm more focused on biomedical things, right? That's why actually I'm talking about this. Evo2, it is a DNA sequence alignment free model. Then you may ask what is sequence alignment free model, because here you may heard about nucleotides without any proper format. You are just giving that because raw text you are giving to chat GPT, right? The same way nucleopeptides are giving as a without sequence alignment, and it should do the cellular organization, protein, RNA prediction, etc. This is an work by Arc Institute and experts. The Duke University's work, they will convert the cell generation or virtual cell generation they have done. And the Harvard people came with an Apollo multimodality where we are getting age, sex, ethnicity, biopsy, everything. Everything should be fed into the model, and the Apollo should encode and decode, and it will make the predictions. So they have a lot of materials available in Harvard Mahmood Lab. They have their pipeline should be there, because if you are interested to work on the medical imaging, there are so many tools available in their repo that we can reuse. And if you are taking for a histopathology model, you have Uni and Virchow. Uni is made by the same Harvard team, and the Virchow is developed by the Page AA team. These both weights are available in the Hugging Face. From the Hugging Face, you can pull the weight and you can train your own model for fine tuning purpose if you don't have enough computation. And what is the building block of a foundational model? Actually, you should give a biopsy, and the biopsy should be cut into different patches, right? And dispatch is again converted into tokens, like the 14 into 4, 16 into 16 or 14 into 14. And the idea came with a paper, image is worth 16 to 16 words. That is the paper which become the foundation of all the visual foundational models. And the same thing, what we are doing is that we are making it as a vector and feeding to the transformer. That is the what is a novelty? Novelty is cutting the spatial entities into vectors. And these vectors will go through the encoder and you are getting a representation. From this representation, we can do the classification or whatever things you can do. And the VIT, you can see different sizes, like it may be a patch size of 8 or a patch size of 16, 14, or 32. What is the need of these different patch sizes? Because if you want full details, you can go for a patch size of 8. If you want a fine-tuned one, you can go for a 14 size. But if you're not focusing on that much on the Core features, you need 16, and more Cores you can go for 32. But the computation amount you can reduce by reducing the patch size, or increasing the patch size, because patch 8 means number of patches may be increased, so you need more computation. That's why Dino V1, Dino V2, Dino V3, these are the metas models. This can be the foundational model for all type of biomedical works. So at that time, you have to understand what is Dino V1 came with 8, Dino V2, V3 came for 14, and some models you go for 16, and Clip is a model used for 32. So this is another foundational model with the idea of contrastive pre-training. Over, you have an image, you have a text, the image and text will be passed through, and the find the similarity, contrastive laws between the image and text, and you are learning from that. It's called few shoot because you are giving some kind of information to the model. But people may now focus on more on zero shot. Zero shot means you don't have given any information from your data, the model should learn. Even in people were thought like, oh, only clip is there, only DIT is there, what should my work? Now the current works you can focus on like how to get the embeddings to be more aligned, or the embeddings, how much distance should be that. That's in the vector space, we have to analyze the embeddings. It's like so many metrics are available, coherent metrics and so many metrics are available. You can work on like how to get the embeddings to be more suitable. And some data sets should not have that kind of thing. How can you embed that? That's the kind of work we can do on clip. And then in Dino V1, V2, V3, actually we knew that Dino architectures came with a distillation approach. The distillation paper came in 2016 by Hinton Lab, and what they talk is that a teacher model should be there and a student model should be there. The teacher model should be capable of processing a big model. It should be produces something output, and the student model should also make the output, and you can apply a loss function. Whenever we are talking about loss function, it is a kind of research approach where you can find out which data is suitable for which loss. Because sometimes the loss function should be in biology, it should be different, and some in physical world should be different. So the loss function, we have to find out which loss function should give a better way of doing. That is also a kind of research we can focus on. Normally, people will go for a cross entropy or a contrastive loss or some, it depends on the network. But that is a kind of way which loss function will give more accuracy. So in Dino, they came with the same idea, but their idea is teacher and student are of same model, same parameter model. Then you may ask why student and teacher? Because the teacher updation should not done eventually. It will go for some 100 or 20th or 30 epochs only. The student teacher way should be updated, but the student way should be updated gradually. So what is the demerit of Dino V1 is that after a few epochs, sometimes the teacher's quality is vanished. So they came up with Dino V2, the same idea, but they have used a VIT of 14 into 14 size. And what is that? They came up with some other loss function like and other some regularization parameters also introduced, like IBOT loss function that is masked and finding out loss function, that some of the packets of students should be masked, and students should be capable of predicting that from the knowledge which already trained. So that's a kind of loss function they have used, and they have used a polio regularization. The regularization is the embedding kind of regularization. That is how the embeddings are similar. And the Dino V3, it's a newly introduced model because previously Uni work of everything is fine-tuned of Dino V2. If we are going for a new foundational model, the Dino V3 should be the default choice. They came up with all the properties of Dino V2, and they have a new feature called gram anchoring. Gram metrics, sometimes if you have a mathematical background, you already knew that it is a feature similarity matrix. Because after training so many epochs, sometimes the local context lost by the model. That is the disadvantage of Dino V2. Then Dino V3 came up with another loss function called gram anchoring, and that makes the model to learn better. So these models are called zero shot models because you are giving just the image. From the image, they are getting all the information. So while taking a foundational model, either you can go for a clip-like thing, otherwise you can go for a dino-like thing, and more models to come. And because all consisting of the transformer networks only. And here also you can ask like, what is the architectural choices I have to take? This is bit more mathematical because you should understand the internal working of the model, and then you have to find out a best loss function, And you have to understand the architectural choices, like how the model is learning. That is why the reasoning models, if you understand the reasoning, then only you can change the design choices. But even after 2020, there is no more choices, like everyone is using transformers and with different fusing, different layer of things, etc. That is why the you may heard about this DeepSeek. What is their core advantage is that they have applied a latent latent attention, because in attention we have a complexity of quadratic one. So to do that computation, they have only 800 GPUs. So what they have done is that how to make the computation more efficient by using a latent attention, because we already knew everything is in vector space. If you have a 1024 dimension, if you can reduce that to 512 dimension, you can reduce the computational cost. So they have applied that mathematical trick, and their model should be a click in 2025. And now new models came with some other kind of this kind of innovations. Like in medical imaging, PCA and UMAP, they are the both important thing, because you have to visualize everything. So for a people who are working on medical imaging, they should have a lead in clustering, harmonization, and PCA. These are the three important categories of people, because the thing is that if you have to visualize everything, you should understand this thing. And what is a classical pipeline of any medical foundational models? You have a data set, you have to do some QC, you have to do patch extraction, and then patch filtering, then training, then downstream tasks. This is a conventional pipeline, any model which you are going to use. So if you are applying for any domain or histopathology, you have to go through these steps. And some of the models which already in showing in histopathology are Uni and Uni 2, Virchow and Virchow 2, and Projigapath. Projigapath is a huge model. But Uni and Uni 2, Virchow 2 are the models which generally people fine tuning for the better performance in pathology. But what is a huge disadvantage of this generic pathology model is that you have trained for different organs, uh, but the tissue is inherently complex. So if you train a model for all type of data, sometimes it hallucinates. So we want an organ-specific models. That is still a biggest thing. Like the number of data available for a single organ should not be that much huge. So this kind of challenging tasks are there. If you have more data, you can make an organ specific. And this is how Uni trained. You can see the different types of slides, number of slides, and different organs. So the lung should have a more bias towards in Uni, because they have used more number of lung images. Kidney should have 8,000, like that number of slides. But if you take number of slides, if you make it patches, it will go for billions. And the same architecture Uni trained with the Dino, because you can see the masked global crops and local crops and pass through the Uni student and teacher model, and it will go through embeddings, and they have two losses, reconstruction loss and arrangement loss, and they find out the learning algorithm by which the model trained. So this is the architecture of Uni. And they have applied for a different types of downstream. One is retrieval, because if you have a patch, you send to the model should find out other patches of the same model, same things learned. And also classification they have used, and segmentation also they have used. But here also one disadvantage is there, like if you are doing a PhD and you are doing work on kidney, sometimes you have to check the model whether it's work for other organ, and sometimes your model should be perfectly for this organ, that should not be proper for other organ. So that is wide level of opportunity are there in the foundational models. And the design choices they put forward is they took 20x magnification, that is they used 112 microns. Sometimes in case of kidney, they can't able to always capture globally. because it may be a more size of that in the physical tissue space. And they converted to 16 to 16 tokens. You need 2 is to 14 to 14 tokens and a Dino V2 pre-training. But in the pre-training, it's also tricky. Like the global crop and local crop should be there. Local crop for the student, global crop for the teacher. The global crop sometimes may be 190 to 224. You not always reach at 224. Sometimes you will get 190 only crops from the teacher only. So this pre-training makes the Dino a vulnerable one for the some kind of tissue tasks. And they came up with this kind of losses, self-distillation loss and IBOT loss and coderegulation. This coderegulation actually we can think like it is in a unique embedding space how much the patches are communicating. So this is also kind of a regularization. We can apply some other also for Dino V2 retraining. And the workout they are good because they have taken a big patch 392 into 392 and they make it to 14 to 14 tokens and they take a global crop of 224 to 224. They got the fixed 224 224 crop. Why they fixed that for 224 224? That not 390 to 392? Because at that time they have V100 GPUs only. Because the GPU is also a dependent parameter for training. So whenever you are making a model, the model should fit in the CUDA. Like it should have the memory constraint should be there. In case of medical images, even this billion actually training the model should be simple, but the how the big model should be trained, that should be more tricky. And then they go for the Dino V2 pre-training with the same loss functions which you need to also use. So they introduced only one design choice. They have a bigger patch size. So now people can experiment with 512, 512. Experiment with 448, 448. But you should always consider like it should be divisible by 16 or 16 or 14 by 14 because you otherwise you have to add loss to the or the noise to the patch. So this is some of the experiments like in kidney images with Virchow 2 and Uni and the bold X structure is called glomeruli. And inside the glomeruli you can see in the fine-tuned version some more dots. These are called podocytes and mesangials because we are more focused on understanding what is inside the glomeruli. Because if in case of uni, you can see only the green things. You can't see other things inside that one. But in case of fine-tuned one, you can see so many things inside the glomeruli also. So this kind of approach will give better kind of visualization to the physicians. That is why the healthcare need a specific organ-specific models. And you can see this is a kidney biopsy. The left one is a needle biopsy and the right one is a core biopsy. And you can see the red ones, they are called glomeruli. But we are failed to get the what is inside the glomeruli because we fixed the glomeruli in the case of a diseased kidney and a normal kidney. But we are failed to get the inside ones. So even though to get the normal kidney to diseased kidney, we can easily identify with the same artifacts like the same model. It will predict the normally the same colors, but still we fail to get what is inside that. So what is the approach? That should be more tricky. Like we have to find out our own models to train the thing. Then it will find out better ingredients in the inside the glomeruli. And then people go for like all foundational models are working like to get segmentation classification. Suppose we have in a biopsy, uh, it has some problems. Even we knew that all problems are due to genetic factors. So can we understand what type of genetic expression from a biopsy? Even now also tools are there like Cinium, Visium. They are high level scanners. Like they cost huge money should be needed for doing that. So instead of that, from a biopsy itself, if a translation researcher can understand what should be the genetic expression or the RNA count, then that should be tricky. That is called spatial transcriptomics. So there are two methods like VCM, that's called sport-based, and a CNEM, that is cell-based. CNEM is more costlier. VCM is because VCM actually many cells share one spot, so there may be number of expression should be less. CNEM, a single cell will get the RNA sequence. So what is the connection? You have a whole slide image, you have a gene expression data, you pass both through the network, and you are applying a cross attention, and you will get a joint representation. And after that, what you are doing is that a biology aware loss functions we can apply. That may be a contrastive or a survival classification or reconstruction. And these losses, if you pass and fed the network, finally your network learned. Then if you pass an HN image, you will get the the genetic expressions. This is a trending one. And but the huge problem is that even if you are getting the genetic expressions, sometimes for the ground truth, there may be also noise. So we need a neural network that should study the uncertainty, some kind of probabilistic neural networks we need for processing that. And this is kind of some of the examples, like you can see this the left side one is an HNE, and this right one is a nuclear gene expression on the data. And the data is the expressions are labeled with respect to different colors. That genes are called, you can see from the like ALD, ALD, OB, TP, ION, etc. So this is what a foundational model can do. And as we go, you can see one more thing, like the nucleus. Actually, we try to fix the nucleus. Then we are just finding about what are the genes present in the nucleus. And that may be more like there may even the CNM scan itself, they are running algorithm to detect the nucleus with respect to HN image like color. Even in that case also, there may be staining a problem, because here we have used data with the HNE only. But if you go for some other data, how to fix that? That should be a more challenging task. And whether will agents help? Yes, of course. Actually, this is a biopsy we have given to Kimi. It's an open model, and we got some good kind of predictions, like this is a normal renal parenchyma. So like that, it will detect. So what happened is that we have enough images, and we don't have any like manually annotating is difficult. So we took all the images and fed to a model like Kimi, an open source model, and it predicts something. And we can do a quality checking, like whether the biopsy is good or bad with respect to its prediction. And we can just pass that to a pathologist, and we can just finalize whether we can use or not. Even the general language models also good in doing things, but the inherent complexity will make them to make confuse. So this is one of that idea. And people who are working for this one, you can go for TCGA, GTEX, SITAC, and HEST1K datasets for developing their things. But more number of data are available, but that's lab specific. And people who want to do starting point as a biomedical one, they can go for these kind of data sets. Downloading that turned to be a tricky thing. Like organ specifically, you have to find the manifest file, and you have to do the processing like that. The HEST1K is a open source spatial transcriptomic data set for VCM. That's developed by Harvard at Harvard Science, and actually you can use that one. And so many CVPR papers came with this idea. But what is the difference between a CVPR paper and a Nature paper is that the Nature paper you will get methodologies and it's biologically significant. CVPR is more technically significant. So if you can fuse two things together, you can make a big project like that. So the conferences like A-star conferences you have to follow in order to get a good idea of biomedical, how the new technology, how new techniques can be applied. And for a better biological meaning, you want to get from Nature papers. So if you collaborate both two, we can do something much better. And what are the computational questions as a kind of computer science we can ask, like the matrix multiplication? Of course, Kublas is done by the NVIDIA, but we can customize more better. Like matrix multiplication, we can reduce the complexity by utilizing more number of cores. Quack is a stands for, they have a group called Trido group, then they are working on that one. And we can use how effectively this metrics can be multiplied. And flash attention 4, we heard about attention mechanism, QK transpose by root take in V, but that is not the core idea. Actually, how to make it in a CUDA to be better performed. And also we can go for fully sharded data parallel because you have a huge model and you have number of GPUs. How effectively you can split the parameters into different models and they have to combine. That is also a training techniques we have to use. So these are the core areas where computer people can do better. But in case of CUDA, what happening is that it's NVIDIA's proprietary. So what we can do is that we can understand their libraries and make a benchmark on your GPU, how to do better than that. And more to do, but still there are many kind of things mm, uh, like new libraries, new papers, or also came in this area. But what is the disadvantage is that the data is very like real time data is very noisy. So the quality check and the cleaning is a difficult task. And I think the rest of time you can ask something related to that one because I think my slides will not talk too much. And I don't know how 49 participants I can see in the screen, but I don't know how many people are listening. But still you can ask questions. That's it.
Thank you, sir. Participants, if you have any queries, you can put in the chat box or you can unmute and ask.
And also I can tell something like this area is people who are doing PhD, they can go for this kind of area because this is trending one. Like if you are going for a generic computer vision problem, it's tough to do the things because already big companies are working on that. We will not get that much GPUs. So the trickier one is to apply on this kind of thing. Because even in this thing, if you want to fully train means you need dedicated GPUs. But I think we have to get a V100 also. It should be very difficult to get, even though national computing mission is there. From that, actually, we can get some GPUs and we can train. So if a one has to start on this one, what they can do is that they can start with Google Colab and they can use a pre-trained model and they can just fine tune that one. And with a small data set, if it's working, fine. Then actually you can go for the 1 hour or 2 hour GPU training and they can get some results on a small data set. Because otherwise you can't able to complete within 2 or 3 hours. And also you can work more on pre-processing and that kind of things because that's more important in kind of medical data. And also post-processing also. That is also a like a research trend you can do. And also while reading the papers, go through the data, go through the ablations, and go through the methodology. These three things they you can fix and you can make your own things.
So we have some questions in the chat box.
Okay. You can. Actually, I will one by one. I will start. For a student who is new to biomedical, what learning path and prerequisites should be recommended for understanding and working with foundational models in field? As if you're starting as a student, I don't know, like BTech or what level, I don't know. But actually, if you are starting with a biomedical AI, only it's reached different region, different levels of biomedical we have reached. I think in India we have started only. I think in India there is a huge tremendous scope there. You can start with fundamentals of computer vision. Actually, you can take the course in deep learning for computer vision by Vineet Balasubramanian at IIT Hyderabad. That will give you a foundation of computer vision. After that, you can read some papers from CVPR, ICLR, and ICML and NURIPS. These four conferences, if you follow, you can get the current trend of biomedical. And then what you can do is that fix one a domain. The domain should be something long or something you have to take. And you have to read something about that because uh, without a proper data knowledge, you can't able to apply your algorithms. Then I think you can spend some 2 to 3 months to get a computer vision background, or 1 to 2 months for fixing your domain, and never go for all domains because no one can be good in everything. So focus on a specific domain and work on that one, and find out the labs which are good in that, and just make a communication with them. We don't know. Sometimes they may be interested more in science. Then second question is like, can you suggest some of the research papers from which we? Yeah, I already mentioned that. You just follow nature papers for biomedical. If you are the engineering kind of thing, go for CVPR, ICML, and new RIPS. Because in 2002, and also WSCV is also good. And people who are just want to do something abroad or something, you just focus on writing or reading this kind of papers because this will give you a huge push into the research domain. Because people will tell like AI will break, etc. But there are fundamental studies like how the embeddings are there, what is spacing there. If you're good in mathematics, good in biology, good in computer programming, you have so many opportunities are coming. Even though if you're not that much good, but read about that, then that will give a huge impact. Because I think in US, almost all labs have vacancies on this biomedical AI and genetic sequencing, etc. So you should have a curiosity towards what I have learned and what to do, etc. That curiosity also will make a huge impact. Not about your intelligence, but curiosity should be needed. Uh, what do you see as the next major breakthrough in biomedical AI? Surely reasoning models, because it's already in trend, and the agentic AI. These two things should be the massive thing because if you pull the biomedical AI, what is the disadvantage of agentic AI is that the data is secure and the model should or the agent should do something crazy that should be difficult. So that's a challenging thing. That like a protocol rule should be maintained for the agent in biomedical AI. That should be also a designing choice. So that are the huge things fighting. And also people who are from teaching teachers, like if you want to mentor your students, you can give them the project on this area, like data sets are available. And also if you give a request or something, you can get the data sets from the that open source data set. We can do some benchmarking. Uh, then of course you will get some real world data with some hospitals or something. That should be good, because we are proposing like want to make it the model, because it's very rapid here. We need within 6 months, 7 months, we need the model, because it's a competitive market, because pharma companies are funding and so many labs are working. So it's like a good opportunity for people who want to explore. And it's like going in computer vision, like core area means usually that much competitive. If you want to be like curious and if you want to learn new things, this area is to be too good. Any other questions? I think you can ask the question also. You don't no need to type the question. If you want to means you can ask also, no problem. Uh, any anyone having doubt, they can ask, no problem. There is no need to chat.
Participants, if you have any queries, just unmute and ask. Sir, I think no more queries are
Okay.
there.
No.
So
Okay.
yeah, let's wind up this session, sir. Thanks a lot. Thanks a lot for sharing your expertise with us.
Okay.
Yeah.
Thanks. If anyone having any doubt on this kind of areas, uh, you can get the contact ID from the organizer, and you can contact me and further things. Okay. Thank you for that. Thank you for giving this opportunity, Sripati Institute of Management Technology and the CSC department, for all the support. Thank you.
Thank you,
Thank you.
Jayanthan. Thank you for your
Thank
time. And participants too, thank you. And we will meet the next day for the next session. Thank you. The feedback form and the attendance link is shared in the chat. Please do submit. And thank you all.